Hey, I'm Mark Herman. I'm the CEO and president of AgriLiant Genetics, based out of Indianapolis, and I'm also the chair for the American Seed Trade Association this year. On behalf of the American Seed Trade Association Board of Directors, the members, I really want to express my deep appreciation for all of you joining us today and joining the CSS here in 2016, as well as the Seed Research Conference Expo. We have a great meeting this year with excellent educational sessions and an excellent expo that I hope you'll get a chance to visit. One of the most important issues that we face in the seed industry today, President and CEO. Thanks, Wendy.
Thanks, Mark. It's nice to be able to kick this off, and it truly is an amazing time for the American seed industry with respect to the evolution that we're seeing in technology and innovation, and truly putting it in basic terms, understanding of what plants do and how we look at those characteristics that are favorable to crop production and addressing the challenges that farmers have with weather and pest and disease. But also, how do we deal with those traits that are characteristics that are maybe not so favorable, and how do we minimize those or in the breeding process? So we want to talk to you a little bit about that evolution of plant breeding and where the industry, as well as our partners in public research and our land-grant institutions and USDA are going in that today. So that's really the focus of the American Seed Trade Association as we move forward in the coming years in dealing with policy domestically and internationally. But one of the things that we like to stress in the beginning as we lay this foundation on plant breeding is an evolving practice. It's an evolving science. Whether you go back to the 10,000 years ago when man was selecting plants that were strong, that met the needs that they had for their local area, all the way to the more recent things in the 1900s and the various practices that we see of crossbreeding and hybridization and mutation genesis and cell culture. Plant breeding builds upon itself. It's not all of a sudden a wake-up of some new technique or new understanding. We understand things more and more each year because we're better at mapping genomes and dealing with bioinformatics. So what we get into today that is really what we see as the next evolution of plant breeding is genome editing and the opportunities that come into play there as we go forward. But it's important that we stress that it will be there are new techniques and new understandings in 2 years, in 5 years, in 10 years that will help us be even better prepared to address those challenges that the grower community and agriculture, food and agriculture address in the future. And when we talk about it, we like to talk about plant breeding innovations because there's many different practices that come into play in this and techniques for breeding that have to be highlighted as we go forward. And it's the evolving plant breeding methods like genome editing work within the plant's family, the characteristics that are naturally occurring within a plant family, whether it's the wild relative or whether it's the product that is sold into the grain industry or into the produce market. There are widely diverse characteristics within plants. It just depends on which one we're harnessing to bring into the marketplace to address the challenges or the opportunities for consumer demand. We look at these methods, they build upon as I mentioned, what plant scientists and breeders have been doing for many years. And it allows us to reach the same endpoint as we would through traditional methods, but in a more precise manner, really going after those characteristics that are favorable to the endpoint that we're looking for, and hopefully not bringing in all of those, what I would call deleterious characteristics that we end up having to back cross out, hopefully at some point down the road. And then the relevance to consumers. They continue to want that safe, healthy food supply for themselves and their family, and they're concerned about their environment as well. How do we begin that process of addressing those on behalf of those consumers so as they go through the process of production of that crop, processing of that crop, and then distribution into the food chain, they've got confidence that it started at the very beginning. And then the consumer wants the farmer to produce a variety of food choices, while using those natural resources and inputs wisely and solving challenges locally and reducing crop inputs, or that potential to reduce crop inputs as we go along. And ultimately, it's that relevance to the farmer. The farmer is the one that ultimately makes the decision of what to plant, where to plant it, and what market it's going to be planted for. The farmer's constantly looking to grow more using less. Farmers need that variety of seed choice, solve their local needs, to manage changing weather, to fight plant disease and pests, and wisely use crop inputs and those natural resources as they go along. So ASTA's overarching policy position when we look at plant breeding innovation is pretty much summed up in this statement. I'll go ahead and read through it. Plant breeding varieties developed through the latest breeding methods should not be differentially regulated if they are similar to or indistinguishable from varieties that could have been produced through earlier breeding methods. Currently, our plant breeders across the board, whether you're in flowers and vegetables, whether you're in row crops as we have here, or in conservation crops, they're bringing new varieties to the market all the time, no matter what method they're using in plant breeding. And they're not pre-market regulated or deregulated, as we say in the biotech side. And they were brought to market because we have the confidence, we have the understanding as plant breeders that they are safe for the environment, that they will meet the requirement or the need that the farmer has, and they will perform. And so we've got a many-year history of taking care of that and bringing those new varieties to the marketplace. That's our goal with respect to policy direction into the future. And we are not only looking at it here domestically, we're looking at it internationally. We realize that it's important that the international arena adopts policy that's consistent and that we don't get into a patchwork globally where it becomes market barriers when breeders and seed companies use certain breeding techniques. So we want to ensure that we have that consistent international policy so it doesn't have a negative impact on research collaborations. We have to be aware of the impact on commercial seed trade globally and the impact on trade and agriculture products across borders. We need to determine the range of new varieties for farmers and new products for consumers. That's done globally today in the seed industry. We move seed from North and South America all the time for winter production, counter-seasonal production. We move seeds in the vegetable industry quite often from Asia, from Africa to the U.S. and back out to other parts of the world. So we've got to be able to do that relatively easily through the process. And then the impact on overall innovation in agriculture development. As we've seen in current biotechnology practices and in research, there is a barrier today. There are a lot of institutions, land grants that are not able to employ these techniques because of the regulatory hurdle to research with them. And it would really be disappointing. It would be a shame if that same practice or requirement went into the breeding techniques as we move forward. Ultimately, we want to look at that toolbox. We think the breeder, the plant scientists need to have all of those tools available in order to bring those new varieties to marketplace to address climate change, to address pest and disease pressure, to address other things in order to bring the consumer what they demand and the needs that the farmer has. So with that, Mark, I will go ahead and move on. Thank you.
Hey, thank you, Andy. Next, I'd like to invite Dr. Jerry Flint to give us a few remarks.
Thanks, Mark. I'm going to try to build a little bit on what Andy has talked about in terms of the new tools that we have available for plant breeders. I work for DuPont Pioneer, and DuPont Pioneer is celebrating our 90th year as a company. And one of the things that we've been very focused on since the introduction of the first commercial hybrids in the 1920s is to continue to look for how growers make their seed choices, the technologies that they want, the things that are important to them in terms of as they're making their seed choices for their production systems. Not only do we operate in the U.S., but we operate in over 90 countries globally. So we take this very seriously in terms of how we look at our research and development program and how we bring new products to the marketplace. Since the beginning of our first introduction of commercial hybrids in the early 1920s, we were also involved in the introduction and co-development of Herculex products with Dow AgroSciences, which brought insect control as a new tool that we could provide to farmers in the U.S. as well. And then as we've introduced other products along the way, we've looked at how we can deploy new technologies and tools. As Andy indicated in his timeline, a number of tools and the evolution of plant breeding as we're going forward. And we do that with a focus on not only the near-term future, but what's happening further out. So when we look at climate change, we look at population growth, we look at nutritional changes that need to occur, we need to continue to upgrade our toolbox in terms of what's available to plant breeders so that we provide the best tools in their research and development program to meet those growing needs. One of the tools that we have within our toolbox is CRISPR-Cas. We're a leading company in terms of how we're utilizing CRISPR-Cas for plant applications. And one of the ways to think about it is in terms of what we can do, a tool that we can provide to breeders that allow us to not only do targeted gene deletions, but also edits, and then in some cases search and replace. And one of the things that we do is look at the plant genetics that already exist and how we can deploy those in the latest varieties that we have. And you're going to hear some examples from some of our other speakers and how that application can be made not only in the crops that DuPont Pioneer is interested in, but also other crops that would include vegetable crops as well. One of the things that I'd like to also talk about is we recently signed a collaboration agreement with CIMMYT, and CIMMYT is located in Mexico. It's the International Maize and Wheat Improvement Center. And this collaboration agreement will allow us to work very closely with the collaborator to help address some of the needs that we have in developing countries. And one of the first targets that we're going to work on is the maize lethal necrosis disease. And it's a very devastating disease in Africa, and it can cause significant yield loss to growers. And this is a collaboration that we're very excited about because it will allow us to look at some of the solutions to problems that plant breeders have not been able to address in the past. And so although this is the first product that we're working on, or the first target that we're working on with the CIMMYT collaboration, there are others that we're going to consider along the way. The other thing that we also know that we need to do is listen to stakeholders, so that we know that with any new technology, there is a social license, if you will, that we need to consider as we're bringing new technologies forward. And so one of the things that we're doing as we're developing our products in our pipeline is reach out to stakeholders, listen to them, understand some of the questions that they're asking, and try to figure out a way that we can have a productive dialogue around the value of the tools that we're trying to deploy and the benefits, but also understanding in terms of the tremendous opportunity that this provides for plant breeders. So with that, I'm going to turn it to Corinne, and she'll talk to you about what's happening with the Sakata.
Hey, thank you, Jerry. Next, we'd like to invite Corinne to come up, Corinne Marshall with Sakata Seeds, to come up and share comments.
Good afternoon. I'm Corinne Marshall with Sakata Seed. I wanted to talk a little bit today about how gene editing might be applied in vegetable production. Before I get into that, I want to just talk briefly about Sakata. Sakata is a global producer of high-quality vegetable and flower seed. Sakata America is headquartered in Morgan Hill, California. And we have research, production, sales facilities, and staff located across North America. Our research facilities are located in Salinas and Woodland, California, Mount Vernon, Washington, Fort Myers, Florida, Sinaloa, Mexico, and Las Cruces, New Mexico. Sakata is known to be a global leader in broccoli, but we have emerged and established ourselves and several other vegetable varieties and species, including watermelon, but also spinach, pepper, and tomato, and several other vegetables. But looking specifically at the challenges that we face in vegetable production, viral diseases transmitted by insects can be a huge problem for us. Fungal pathogens in the soil, such as verticillium and fusarium, are also quite problematic. Downy mildew in spinach, foliar diseases, bacterial leaf spot in peppers and tomatoes. Those are just some of the many disease and pathogen-related problems that we're facing. The conventional solutions have been obviously chemical control, which can be applied when needed and can be used to address multiple issues, but that really restricts us from markets such as the organic market, and there are environmental risks associated with chemical use and additional growing costs. Other ways that we've conventionally dealt with or tried to deal with these issues include, just being able to build the resistance into varieties and to be able to, not be restricted to play in the organic market. And there are no environmental risks associated with that, and the protection's there when you need it. But the problem is that resistance, and the way we deal with that is very specific. We look at specific races, specific diseases, and the cost can be quite expensive. So taking spinach, downy mildew resistance in spinach as an example, the demand for organic spinach exploded in the 90s, and since that time we've been challenged with trying to come up with ways to deal with the problem, and we're basically chasing the resistance, and the races pop up annually. And so the future of organic spinach production is questionable, because how long are we going to be able to sustain the ability to keep up with the challenge of dealing with the resistance, and come up with ways to handle that. So gene editing could provide the solution to having a stronger resistance to downy mildew and spinach. Gene editing can be used to enhance the nutrition in many vegetables. So this goes beyond just dealing with disease and pathogen, but enhancing nutrition in vegetables is actually, I think, a very exciting prospect. Lycopene and glucosinolates in broccoli, which can reduce chronic disease or slow disease such as cancer. Sulforaphane is a glucosinolate in broccoli, and most of us know that when we cook broccoli, we lose the nutrient. So gene editing could actually help us solve that problem, extend the nutrient to the cooked vegetable. Tomatoes and watermelon are good sources of lycopene, and so interestingly, in tomato, lycopene is more bioavailable when it's cooked. So to be able to have that nutrition in the fresh product would be exciting. L-citrulline in watermelon helps reduce blood pressure. So to be able to enhance watermelon and increase the amount of citrulline would be quite exciting for us. So compared with backcross methods or traditional backcross methods or mutagenesis, gene editing is more precise and more efficient. But this will not change the way we do things as far as product development is concerned. The rigorous product development process that we go through to bring products to market, to test them and make sure that they're viable. Areas of future plant improvements that we could look to for gene editing beyond disease resistance and nutrition could include increasing the nitrogen use efficiency, or improving drought resistance, flavor, yield, and seed quality. And it's possible that gene editing could also be extended to flowers. So our president has passionately stated that vegetables nourish the body and flowers nourish the soul. Thank you.
Hey, thank you, Corinne. And now, to conclude our comments, Juliette Marshall, Associate Professor, University of Idaho, and then we'll move to Q&A.
So I am a pathologist and an agronomist, and I work at the University of Idaho. And wheat and barley, as you know, are extremely numerous. We can talk about bread, we can talk about pastries, we can talk about, or we can talk about my favorite, which is beer, right? So numerous products, numerous uses for these products, and I'm very passionate about what I do with wheat and barley. The issue happens to be that there's a disease of wheat and barley called Fusarium head blight. It's also called scab, lovely name. But there's a reason it's a scabby disease. It's an ugly disease. Okay, so this is a picture here on the right. You can see that the wheat is in grain fill and it's filling. You should have beautiful, big, fat, plump kernels. And instead, this disease comes in and starts to cut off the production of good, healthy kernels. And so you get scabby kernels, Fusarium damaged kernels, and they're shrunken. You're reducing your yield. But in addition to that, what this disease does is it also coats the outside of the seed, not only with the fungus, but with a mycotoxin. And of course, these specific mycotoxins include deoxynivalenol, 3-adon, and 15-adon, which are derivatives of deoxynivalenol. And this toxin is nicknamed vomitoxin because it causes vomiting. In humans, other monogastric animals, the test subject for tolerance to some of the vomitoxin is actually minks. I can't imagine feeding enough vomitoxin to minks to get them to vomit, but the toxin is limited by the FDA in human food and products to be 1 part per million. Now we're seeing this vomitoxin showing up in a lot of our grain, whether it's for pasta, whether it's for bread, whether it's for barley, for beer. We're seeing large levels of this toxin, and in fact, I have seen over 25 parts per million in some durum wheat in Idaho that had to be thrown away. Okay, so there's a lot of limit to the contaminated barley and wheat as to what we can do with it. We can try to feed it to some of our livestock, but again, it's also toxic to a lot of our livestock. So this toxin is something that is, in addition to yield loss, something that we are very concerned about in our products and our product development for wheat and barley. So I want to bring the subject to bear about management of this and what we can look to in the future. This is a bad disease. Once it gets going in an area, it requires kind of an all-hands-on-deck approach to try to reduce it or control it. We use crop rotation to try to reduce the residue of the wheat and the barley. We try to use crop rotation to get away from corn because corn is a good source of this fungus to grow and reproduce. So it requires residue management, it requires planting date management, it requires fungicides. So application timing is critical, and the exact type of fungicide is critical. And we have a scab forecasting methodology in the Midwest that we utilize to try to give warning to our growers. Okay, now the scab risk is high. You have to spray, and you have to spray exactly in this little window to try to get maximum control of this disease. But even with all of these practices, we don't have the host resistance to actually control this disease. So when the conditions are right for this fungus to take off, boom, we don't have the host resistance, the environment's right, we have the disease infecting our crop, and we have accumulation of toxins in our grain. So it is serious. So when the conditions are right, even the appropriate variety with the maximum amount of resistance that we have available, even with our forecasting saying, even if you have a moderately resistant variety, you need to spray, sometimes we still don't control this disease. It is a very difficult situation. So this is where I think gene editing could actually come into play to help us reduce the impacts of this disease. Now, first of all, there are no genetically modified wheat or barley varieties or products on the market. There are none. Okay, there's none produced in the U.S. So any product that claims to be GMO-free, being wheat or barley derived, all of our products are GMO-free currently. But I am a proponent of genetically modified crops, and we'll talk a little bit more about that. But what I want to do is talk about the potential for utilizing to reduce this particular disease. Now, the whole plant is biochemical, pathways of resistance, pathways of susceptibility. And what I want to look at in particular is what happens with gene editing and how we can utilize that to try to reduce the impacts. So on the right here you have a typical cartoon of a gene, of the DNA, okay? And what happens with gene editing potentially is what we can do with gene editing, with the CRISPR-Cas system. Say we want to come in there and we have a very targeted spot where we know what that gene is going to be used to develop. Say susceptibility is it's going to be a specific recognition model in the plant. So say we can use that and we can target that spot and we can cut it out. And this is a double-stranded cut, and then the plant says, oh, wait a minute, we need to repair that. So there's a double-stranded cut. It has no template to match that cut. And say we pull out the AT and the plant's trying to fix it and it puts in a different base pair, a CG, a different base pair, then actually rearranges that chromosome. So CRISPR-Cas can also be used to not add the GC, but let's just cut that, those two strands. Let's just cut that, and let's pair it back together. Yes, this, okay? We're all very familiar with this nursery rhyme, and there's a reason why, in my simplistic mind, this is the best way that I could think to kind of explain this, because after doing this, I'm like, yeah, that works for me. Okay, so yeah, Baba Black Sheep, have you any wool? Yes, sir, yes, sir. 3 bags full. 1 for my master, 1 for the dame, my dame. And there comes CRISPR-Cas. We're going to cut it right there. We're going to take out that A, and guess what happens? We've just shifted. You now forgot what those last 2 sentences. You can't even read it, right? That's what we call a frame shift in genetics. It's still all there. If you look at it, it says the and is gone, but now it says 1 for the little boy who lives down the lane. But it's not readable to our eyes. You now can't see that. Well, the same mechanism can be applied when you have a frame shift cut CRISPR-Cas system. Now, the reason why I want to show you that is because now I think I can better explain the fact of susceptibility and modifying how that plant would recognize a pathogen toxin. So this is a very simple cartoon of a plant cell. And what we have at that top is a protein that is that is across the plasma lemma in a cell. And what it does is it recognizes a specific toxin from Fusarium head blight. It will actually take that toxin in. When then it'll start to work on the nucleus and the cytoplasm and the mitochondria and start to destroy the inner workings of that cell. But say we use CRISPR-Cas to mess up that protein, and now it's a skinny little protein and it doesn't quite recognize that toxin. So now we have prevented that toxin from starting the infection process, from facilitating the infection process of that fungus, and it slows down the disease. Okay? So, I think CRISPR-Cas can be very helpful, even though we don't have genetically modified wheat and barley. we can use the techniques to actually either understand the process and then use gene editing to reduce the impacts. So we're all looking at agricultural and environmental benefits. Breeding is first the search for resistant parents. We need to breed for resistance in our crops. Well, in head blight, we don't have true resistance. We have partial resistance, and there are no silver bullets. We want to be able to use gene editing to effectively enhance what small resistance factors are there. Genetic engineering, we've been able to use that as a scientific tool to advance the understanding of the process of infection and susceptibility and resistance. We can make rapid progress with small yet effective changes, and we can utilize novel genes that were unattainable by traditional plant gene breeding. We can modify genes and we can stack multiple genes, but wait a minute, let's look at gene editing. Again, we can make rapid progress with small yet effective changes, but we don't have to do the introduction of novel genes. We don't have to introduce novel genes at all. We can modify the existing genes to enhance those traits which may otherwise be difficult to achieve through traditional methods. And it's really important that the public institutions actually utilize these techniques. The public sector often is where things start. We can provide proof of concept, proof of where things really work. So we can use the transgenics to identify how things work, and we can use gene editing, which is not genetically modified, to then go in and actually produce a crop that would be resistant to the disease and resistant to the accumulation of these toxins. So we're working to enhance those traits and those crops that are also not commercially viable. A lot of this technology has been utilized in some of the bigger crops, corn and soybeans. But we have a lot of crops that won't have that investment from private companies. We can do that at a university level with these techniques. And so, of course, environmental and agricultural safety is our primary priority. So, thank you.
Thank you, Dr. Marshall.